The overall goal of this procedure is to simultaneously assess multiple potential phosphorylation sites and to identify sites for subsequent validation by phosphorylation site mutants.
This method can help answer key questions in the biochemistry and cell biology fields about the role of post-translational modifications in cell signaling.
This technique allows easy, budget-friendly preliminary screening for phosphorylation sites in a protein of interest.
The implication of this technique extends toward the diagnosis and therapy of diseases, including inflammation and cancer, because phosphorylation is an important biochemical event in signaling pathways in human diseases. Aiding the demonstration will be Shin, a graduate student from my laboratory, who will perform the site-directed mutagenesis.
First, combine 50 nanograms of the plasmid PGX GST NRF2, 125 nanograms of forward primer, 125 nanograms of a reverse primer, 1 microliter of DNTP mix, 2.5 units of PFU DNA polymerase, and 10x reaction buffer on ice. Add sterile distilled water to bring the volume to 50 microliters. Perform PCR to incorporate the mutant strand, and then cool the sample on ice for 2 minutes. Add 1 microliter of DPNI restriction enzyme to the cooled sample. Mix the sample with gentle pipetting and then centrifuge the sample for 1 minute at 13,000 times G.